Welcome back, and one more to go, and that would be our friends the alcohols here. So as we said before, what makes an alcohol is this hydroxyl group right here. And that was, let's try to sketch it again for you here. There's carbon, oxygen, hydrogen, and lone pairs. So it's this group right here that makes something an alcohol. Now, are these bonds polar? Yes, they are, especially this oxygen-hydrogen bond. So by definition, so if you look at this alcohol, you've got two really polar bonds, the oxygen-hydrogen, and the carbon-oxygen bond. Everything else is nonpolar, but you've got at least two polar bonds in these, so the molecules are pretty much polar. But these are special, and this is something called, imaginatively enough, hydrogen bonding. And this is and you will note the quotes here, a quote-unquote bond between the hydrogen atom and a very polar bond, like our OH bond, and the unshared electron pair of a electronegative atom that is nearby. Now, this is such a rare occasion, and this polarity needs to be so great that generally you only see these with HF, HO, or HN bonds. So just remember those three: fluorine, oxygen, and nitrogen. Now, the actual interaction is usually between a hydrogen atom of a nitrogen, oxygen, or fluorine of a different neighboring molecule. So here's what this looks like. I'm using hydrogen fluoride as an example because it's one of the simpler cases. So consider a lone hydrogen fluoride or hydrofluoric acid molecule. So this is the fluorine and this is the hydrogen. Now this is a covalent bond, but barely. So remember, fluorine is the most electronegative element. It does not share electrons nicely. It's the electron hog of the periodic table. So most of the electron density in that single bond between the two is actually around the fluorine atom. But recall that if you did the Lewis structure, that one bond, that one shared electron pair, is actually the only electrons that the hydrogen has, which means that if all that electron density is skewed way over toward the fluorine end, that this hydrogen atom is essentially a proton out by itself. It's got no electron density around it at all, which would make it very attractive to a neighboring fluorine atom of NHF, because this thing is, so here we have essentially a proton. This fluorine atom is essentially one big ball of electron density. It itself has three unshared pairs and most of the shared pair between the two of them. And that makes the interaction between this hydrogen and this fluorine incredibly strong. Now, from what I've described, and to the point where some of the electron density here can be shifted over to that hydrogen atom. Now, or not. Now, in some cases, you might wonder why this isn't just a very extreme form of the dipole-dipole interactions that we talked about before. This has a very large partial positive charge. This has a very large partial negative charge, and same thing over here. So why is it different? Why is it not just the strongest of the dipole-dipole interactions? And a couple of reasons. Number one is that they are a lot stronger by about a factor of 10. So these clock in at about 15 kilojoules per mole. And for reference, the dipole-dipole interactions usually are about, somewhere between 1 and 2, maybe 3. So they're a lot stronger than any other type of dipole-dipole interactions. So much so that they are seen even in the gas phase. So if you look at hydrogen fluoride or hydrofluoric acid in the gaseous form, you actually still don't see a whole lot of lone HF molecules. Most of them are paired with one other HF molecule, or sometimes even groups of 3. And that is not the case with other polar molecules, that those interactions are completely destroyed in the gas phase and they don't pair up or triple up or anything like that. So that's different. They also seem to be much more directional than any other intermolecular forces. They tend to align themselves in a very specific way, which is not the case with dipole-dipole interactions or induced dipole or London dispersion forces. And when looked at that way, people think, well, especially with the fact that these are that they're effective in the gas phase and that they actually have some directionality to them, well, maybe they're actually maybe they're not a strong dipole-dipole interaction. Maybe they're actually a weak bond, and they actually aren't an intermolecular force, but they actually are just a straight-up bond. And to this day, people are actually still trying to debate this, and something it actually seems to be a hybrid of the two. And I will point out that this one particular type of intermolecular force, of intermolecular attraction, is considered to be quite important, to the point where it's actually responsible for life as we know it in actually more ways than one. Well, 2. So what I want to do now is give you a couple of examples of hydrogen bonding just to kind of show you how they work with different molecular structures. So here's a bunch of them. So here was the first one, hydrogen fluoride. And again, the dotted line here represents the hydrogen bond. And here you can see the hydrogen with a very large partial positive charge being quite attracted to the fluorine with a whole bunch of unshared electron pairs around it. Now, it turns out that the hydrogen bonds, if you look at this ammonia-water example here, these interactions can be between atoms of different types of molecules. So remember, the key is that the hydrogen has to be bound to a nitrogen, oxygen, or fluorine. So all of these are candidates, and one of these hydrogens could actually hydrogen bond to the oxygen of a nearby water molecule. The hydrogens in water are also capable of hydrogen bonding, and they are perfectly happy hydrogen bonding to specifically the unshared pair of a neighboring nitrogen atom on a neighboring ammonia molecule, for that matter. And the water ethanol example, I want to point out, so again, oxygen and water, both hydrogens are bound to an oxygen. They can hydrogen bond. And water hydrogen bonds quite nicely to ethanol. So this is again the alcohol that we've been talking about. And the hydrogen that is attached to the oxygen can hydrogen bond. But in this case, it's the hydrogen atom from a water being attracted to the oxygen atom, and more specifically, its unshared pairs. What I want to note is that not every hydrogen can hydrogen bond. So for example, the other hydrogens in ethanol are bonded to carbon atoms, so they don't count. Only this one could. This one could hydrogen bond to the oxygen of a neighboring water molecule or another ethanol molecule. Now sometimes, so looking at formic acid here, formic acid is the active ingredient in ant venom. And it's an example of 2 molecules of the same compound being able to make a double hydrogen bond. So if you look at this structure right here, you've got a hydrogen bonded to a carbon, so that one doesn't count. You have the double bond oxygen here, the singly bound oxygen here, and then this hydrogen. So this hydrogen can hydrogen bond and would be perfectly happy lining up with the oxygen atom on a neighboring formic acid molecule. The hydrogen on this formic acid molecule on the right one can also form a hydrogen bond with that oxygen atom on the left one. And in some cases, so this is multiple hydrogen bonds. Now in some cases here, some molecules can actually form hydrogen bonds within themselves. So this is getting into organic chemistry a little bit, but so this is a ring of 6 carbons, and it's completely flat, and there's like double bonds in there. Now it turns out that carbon atoms, actually carbon, oxygen, and nitrogen atoms, more or less together, like to make, if they're forming a ring, it's best if the ring has 5 or 6 members in it. More or less than that, and the angles are not quite right. And in this case, so we've got here a carbon, an oxygen, and a hydrogen. So this hydrogen could hydrogen bond. And it turns out that this oxygen atom here ends up being in about the right spot, that these could actually form a closed 6-member ring. And there's a pretty decent interaction between this hydrogen and this oxygen right here. Salicylic acid, by the way, is a precursor, or is the very, very ancient version of aspirin. It is a compound that is found particularly in willow bark and willow leaves. And it has the analgesic properties, so it's got the same pain relief and fever relief properties that aspirin has, but it doesn't taste as bad. Aspirin is acetyl salicylic acid. It's got a couple of things bonded onto this ring. And again, it makes it a little more palatable. And if you've ever tasted aspirin, that might give you some idea of exactly how nasty this stuff tastes. As a further aside, dogs don't seem to mind it, and it's kind of in a folktale that older arthritic dogs will seek out willow trees to chew on them because they get some pain relief from this compound. And last but certainly not least, I've got glycine here. So this is an amino acid. It's got a whole bunch of hydrogen bonding capabilities. So you've got these hydrogens attached to this nitrogen, could hydrogen bond. This one could as well. And this is a matter of if they line up just right, you can actually get multiple hydrogen bonds. So you've got this NHO alignment here, and then a mirror one, NHO here. So amino acids can hydrogen bond together. And this leads into reason number 1 why hydrogen bonding is responsible for life as we know it. And that's because hydrogen bonding is how base pair recognition works. So you may or may not recall from biology that in DNA, so this is a model, so remember DNA is actually, a DNA strand is actually 2 covalent molecules that are held together in a very specific way because of hydrogen bonds. And you may have been told that when these 2 molecules kind of coil together, that adenines always pair up with the thymine and cytosine always pairs up with a guanine. So here's how that looks. So this is the sugar phosphate backbone. And so here are the base pairs. So this is adenine starting from right here. And then thymine is this thing right here. So thymine and adenine form a double hydrogen bond. Cytosine and guanine form a triple hydrogen bond with each other. And the spacing from these is actually, it might not look like much. So this spacing here is a little bit bigger than any one of these 2, but it's enough to make an adenine utterly incompatible with either guanine or cytosine, and likewise for all the other possible combinations. Only adenine and thymine line up like this to form this hydrogen bond to actually form that link with the neighboring ring. And cytosine and guanine, again, are the only ones that line up with each other, so that's why they always pair up with each other. So this is how the base pair replication works, that because of the size and shape of these molecules, only these 2 hydrogen bond to each other, and then only these 2 hydrogen bond to each other. And since that's how we, copy genetic information, that's kind of important for life. The other one is a little more esoteric. So hydrogen bonding in water actually is not present in the solid. The liquid water hydrogen bonds, but the ice does not. And that's because we talked about the directionality of the hydrogen bond. The structure of solid ice is a little different than what the loose structure that it forms in a liquid. And the angles are such that the hydrogen bonding interaction actually isn't there in ice. Which means that for a small range of temperatures, the intermolecular interactions are actually stronger in the liquid than they are in the solid. And this is unique to water. Remember, I think I've said in class before, water is weird. And this is why water is the only substance known for which there's a temperature range where the liquid is actually more dense than the solid. So the intermolecular forces are actually a bit weaker in the solid, which means the average distance is a little bigger. So it has a higher molar volume, so the liquid is more dense than the solid is. And so this is a picture of the density of water with temperature. And it actually peaks about here. And that's about 4 degrees Celsius. And so that is where water is at its most dense, and at that temperature it is also a liquid. Again, this curve right here is very uncommon. Everything else, the solid is always denser than the liquid, no matter what the temperature is. But this has one very important large scale effect. That if it gets cold enough where you live, you can go ice fishing. Actually, it's not just for the fish. So the solid is less dense than the water, so it floats on the liquid water. Now everything else, the solid is more dense, sinks to the bottom. So ethanol ethanol cubes in liquid ethanol would sink, they would not float. So contemplate a harsh winter if you can. So the air gets cold. The cold air cools down the liquid water and it freezes. But it stays on top. And if it gets colder, that layer of ice gets thicker, but it still stays on top. And unless it is a very, very, very shallow lake, there's always some part of the lake never completely freezes. So the bottom stays liquid, which means that the aquatic life, so the plants and the fish in there, don't themselves freeze. They survive to live another spring. Now, if water was like anything else, what would happen is in the winter it gets cold, the top of the water freezes, it sinks, and then more fresh liquid is still exposed, it freezes and sinks, and presumably never gets, and if it doesn't warm up enough, you're just continually making ice, it's sinking to the bottom, and then you're continually freezing more water, which continually sinks, and it would be very easy for even very, very deep lakes to completely freeze, which would kill the life in it. And it seems like life on this planet started aquatically, and if the lakes and oceans were to freeze permanently, this would have been a problem. But because of hydrogen bonding, liquid water is a little more dense than solid water, so lakes don't freeze, and life survives. All right, we will see you next time. Take care.